In the name of God, in the third part, we want to speak about the inflammatory response. You know that the major way of innate immune response to the infections is the inflammation. Inflammation means the accumulation of leukocytes, plasma proteins, and fluid derived from the blood at an extravascular tissue site of infection or injury. We have two types of inflammation: acute inflammation and chronic inflammation. Acute inflammation can develop earlier after the pathogen enters the body. Acute inflammation can develop in minutes to hours and last for days. Chronic inflammation is a process that takes over from acute inflammation if the infection is not eliminated or the tissue injury is prolonged. In inflammation, we have different processes. We have capillary widening. This capillary widening could increase blood flow to the site of inflammation. We have increased capillary permeability, and this increase could induce the release of fluid from the blood to the site of the inflammation. We have the attraction of white blood cells to the site of the inflammation, and then these leukocytes, these white blood cells, migrate to the site of the injury. And also we have some systemic responses in inflammation, such as fever and proliferation of white blood cells. All of these processes could induce heat, redness, tenderness, swelling, and pain at the site of inflammation. At inflammation site, the first population of white blood cells that enters to the site of the inflammation is neutrophil, and after that monocyte migrates to the site of the infection and tissue injury. So the recruitment of neutrophils and monocytes to the site of infection and tissue injury is the most prominent features of inflammation. The adhesion molecules play important role in this recruitment. Selectins are plasma membrane carbohydrate binding adhesion molecules. They are attached to carbohydrates, and they mediate the initial step of low affinity adhesion of circulating leukocytes to endothelial cells lining post-capillary venules. So in first steps of inflammation, the selectin can initiate the adhesion of leukocytes to the endothelial cells that cover the post-capillary venules, and this adhesion has low affinity. So this attachment can be detached easily. Endothelial cells express two types of selectins. These are pre-selectin and E-selectin. We have different types of selectins: P-selectin, E-selectin, and L-selectin. P-selectin and E-selectin are expressed on the surface of endothelial cells. P-selectin is stored in cytoplasmic granules of endothelial cells, so it can rapidly distribute on the surface of endothelial cells. And the other molecule of selectins which are expressed on the surface of endothelial cells is E-selectin. E-selectin is not stored, it is synthesized and expressed on the endothelial cell surface within 1 to 2 hours in response to interleukin-1 and TNF, which are produced by tissue dendritic cells or macrophages in response to infection. So the P-selectins are pre-stored, so rapidly respond at the site of inflammation and release and express on the surface of endothelial cells. But E-selectins are not synthesized in normal condition, and after the production of interleukin-1 and TNF-alpha with the dendritic cell and macrophages in response to pathogens, the synthesis of E-selectin initiated, and it expressed on the endothelial cell surface. The ligands of P-selectin and E-selectins are carbohydrates that are related to Lewis family ligands. Another type of selectin is L-selectin. L-selectins are expressed on the surface of leukocytes, but they are not present on the surface of endothelial cells. The ligand for L-selectin are some kind of sialomucins which are expressed on endothelial cells. The expression of sialomucin is induced by cytokine activation of these endothelial cells. So if we want to categorize, E-selectin and P-selectin are expressed on the surface of endothelial cells, but L-selectins are expressed on the surface of leukocytes and can bind to sialomucins on the surface of endothelial cells. The other group of adhesion molecules are integrins. Integrins are cell surface proteins that mediate the adhesion of cells to other cells or cells to extracellular matrix. And this ligation is mediated by interaction with various ligands. Integrins usually have two chains, alpha and beta chain. Two integrins are expressed on leukocytes, which are more important in inflammation process. The first one is leukocyte function associated antigen 1 or LFA1, and the other one is VLA4 or very late antigen 4. integrins rapidly increase their affinity for their ligands in response to intracellular signals, which are induced in all leukocytes by chemokine binding to chemokine receptors. As you see, after initiation of inflammation, some cytokines which are called chemokines are produced. These chemokines attach on the surface of endothelial cells, and these chemokine can bind to chemokine receptors on the surface of neutrophils or monocytes, and this interaction of chemokine with chemokine receptor can convert the integrins from low affinity molecules to high affinity ones. So the interaction of chemokine and chemokine could induce the increase in affinity of integrin for its ligand. And this high affinity integrin interaction could provide stable attachment of neutrophils after beginning of the inflammation. If we want to speak about the steps of inflammation process and recruitment of leukocytes to the site of infection, as you see in the picture, you see a blood vessel and a tissue. When a microbe enter a tissue, for example, a macrophage could phagocyte the microbe, and after phagocytosis, the macrophage stimulated by microbes. These macrophages could produce some inflammatory cytokines such as TNF and interleukin 1, and these inflammatory cytokines could induce the expression of E-selectin and P-selectin on the surface of endothelial cells. You remember that I explained the P-selectin is the first one which is expressed on the surface of the endothelial cell, and after 1 to 2 hours E-selectin is expressed on the surface of endothelial cells. Leukocytes express selectin ligand on their surface, and the interaction of selectin ligand with E-selectin and P-selectin could induce the rolling of leukocytes such as neutrophils and monocytes on the surface of endothelial cells. The affinity of selectin and selectin ligand is low, and this interaction of selectin and selectin ligand could be detached by blood flow. So the leukocytes roll on the surface of endothelial cells. At the same time, the macrophages could produce chemokine, and you remember that I explained that these chemokines pass through endothelial cells and attach to the surface of endothelial cells. The interaction of integrin, the interaction of chemokine with the chemokine receptor on the surface of neutrophil could induce higher affinity states in integrin interaction. Here you see at earlier phase the affinity of integrin for its receptor is low. We have low affinity state, but after the attachment of chemokine to chemokine receptor on the surface of the neutrophil, low affinity state of integrin change to high affinity state. So this neutrophil or this monocyte could attach stably on the endothelial cells. So by a high affinity integrin attachment, we have a stable adhesion of the leukocytes to the endothelial cells. And after that, this neutrophil could pass from the space between endothelial cell or transendothelial cells to the site of the presence of the microbe, and this attraction is mediated by chemokines which are produced at the site of the inflammation. So in the inflammation we have different stages. The first one is margination. After the margination we have rolling, and this rolling is mediated by selectins. After that we have integrin activation by chemokines, and after converting integrin from low affinity state to high affinity state, we have stable adhesion of leukocytes to the endothelial cells. And finally we have the migration through endothelium to the site of the inflammation. So in inflammation we have selectin mediated rolling of leukocytes on endothelium, chemokine mediated increase in affinity of integrins, a stable integrin mediated adhesion of leukocyte to endothelium, and finally transmigration of leukocytes through the endothelium. Leukocyte accumulation in tissue is a major component of inflammation. It is typically elicited by microbes, but it may be seen in response to a variety of non-infectious agents as well. I put some questions on Navid webpage, and please reply to the questions on Navid. Thank you very much.